Good morning and welcome to this first live scientific session of the I4C annual webinar that I'm really happy to chair. And we had in mind to have this meeting in person in Turin. So I just want to show you some pictures of our city, hoping that there will be soon other opportunities to meet and maybe have the meeting in person another time. So I don't want to steal time from the speakers. We have 3 presentations today. I just want to give you some information. There will be 3 presentations. These will be recorded and will be available on the website of the meeting. After each presentations, there will be the opportunity for a brief discussion. And all attendees can just type their questions in the question and answer panel of the WebEx screen, or if they want to speak, they can ask to have their microphone unmuted, again writing in the Q&A section, and then we'll allow you to speak to the presenters. So I would start by leaving the floor to Jiangrong He, who will talk about recent findings from I4C, maternal infection and risk of childhood leukemia.
Hello, everyone. I'm Jeff. I work with Terry in Oxford on the topic of maternal infection and childhood leukemia, which I'm going to present. It is a great pleasure to present my project in a live session. One of the molecular characteristic of childhood leukemia is fusion gene, which combine 2 separate genes incorrectly and lead to abnormal gene expression and cell function. Previous studies have shown that most of these leukemia-related fusion genes were present at birth. So this suggests that prenatal factors might play an important role in the development of childhood leukemia. Recently, a few nested case control studies found that the leukemia cases which were identified in childhood had different level of cytokine at birth from those in healthy controls. So this suggests that the immune-related factors during pregnancy might change the immune system development of the fetus, which might be linked to the development of childhood leukemia after birth. And maternal infection is one of these suspected factors. Maternal infection could affect fetal health by different ways. And firstly, maternal infection can reach the fetus by ascending from vagina or transmitted from the placenta. And there are a lot of pathogens that are related to fetal health. For example, CMV, rubella, and herpes simplex and others were a classical group of teratogenic pathogens. Other bacteria could also lead to inflammatory responses in the fetus, which in turn could result in fetal death, preterm birth, or fetal organ injuries. So how could maternal infection impact the risk of childhood leukemia? And this is my hypothesis. The infectious agent could be transferred from the mother to the fetus and then lead to genetic lesions or chromosomal modifications, which were linked to increased risk of childhood leukemia. And another possibility is that the immune responses locked the infectious agent themselves could be transferred from the mother to the fetus and change the immune system development of the fetus. And then after birth, the child might have abnormal immune response to infection during childhood. And this is hypothesized risk factor for childhood leukemia. Another pathway could be related to perinatal outcomes, such as birth defect, cesarean section, or preterm birth, maternal infection could increase the risk of these perinatal outcomes, and all of these have been reported to be associated with higher risk of childhood leukemia. So it is possible that a specific type of infection or infection in general might be related to the development of childhood leukemia. And my research question is maternal infection during pregnancy associated with childhood leukemia? So first, we performed a systematic review and meta-analysis to summarize what have been found in literature. We identified 32 articles from 20 studies, including 15 studies on ALL and 14 studies on any childhood leukemia. And this is the main result of the meta-analysis. We found 12 types of infections were investigated in the literature, and influenza was associated with higher risk of childhood leukemia in the meta-analysis. The pooled odds ratio is 1.77. Rubella and varicella were also associated with higher risk of childhood leukemia, but the number of studies was small and the confidence interval of the effect size were wide. So in the literature, we found influenza, varicella, rubella during pregnancy might be associated with higher risk of leukemia. But most of previous studies were case control studies, and there were very few prospective cohort studies in the literature. And next, we use the I4C data, cohort data, to reanalyze this association. Currently, we have 7 cohorts in I4C, including the CBP from the US, LSPEC from UK, and MOBA, Norway, DMBC, and another one in Italy, and Israel, and Australia. But when I started my project, we have only data on 6 cohorts, and totally we have more than 300,000 mother and child pairs in I4C. There were more than 500 cancer cases, including 167 childhood leukemia. This table shows how the maternal infection data were collected in each cohort, and we can see that most cohorts used self-report questionnaires or interview to collect the infection data. And the timing of data collection might be a little bit different across the cohort. And this is the list of the infection variable which were collected at least in 2 cohorts and could be harmonized across the 6 cohorts. And the prevalence of respiratory tract infection is highest among all these infection variable, followed by cold, influenza, vaginitis, and urinary tract infection. We also have data on 2 infection-related symptoms, that is diarrhea and fever. The primary outcome is any childhood leukemia, and secondary outcomes were ALL and AML. Most cohorts collect cancer data through linkage to the cancer registries. I use multi-level cost model to investigate the association between the maternal infections and childhood leukemia. All models were adjusted for maternal age, education level, parity, diabetes, BMI, and smoking during pregnancy. The missing data were imputed using multiple imputation. So this is the main result. I have 3 models, unadjusted, semi-adjusted, and fully adjusted. But the results are very similar between these 3 models. So let's just focus on the fully adjusted model. And we can see that urinary tract infection and its main subtype, cystitis, were associated with higher risk of childhood leukemia. The hazard ratio is 1.68. Influenza and respiratory tract infection were also associated with higher risk of childhood leukemia, but the confidence interval for influenza was wide. Other infections such as common cold, vaginitis, diarrhea, fever were not associated with high risk of childhood leukemia. And then I stratified the analysis by the subtypes of leukemia. And we can see that the pattern of association between infection and childhood leukemia were very similar to the main analysis. For example, urinary tract infection and cystitis were also associated with higher risk of ALL or AML, but the confidence interval were wide because of the reduced sample size in the subgroup analysis. And again, respiratory tract infection was associated with higher risk of ALL and AML. Next, I investigate whether the exposure timing, the infection timing, might impact the association between maternal infection and childhood leukemia. And I found that the association for maternal infection in late pregnancy, it appears to be stronger than the one for infection in early pregnancy. For example, the hazard ratio for urinary tract infection in late pregnancy is 1.9, but the one for infection for UTI in early pregnancy is 1.2. I also did several sensitivity analysis, for example, adjusted for the birth season or excluding baby born preterm, or just include the complete case data, but the results were very similar to the primary analysis. So using cohort data, we found that urinary tract infection and respiratory tract infection during pregnancy were associated with increased risk of childhood leukemia. And this is the first large-scale prospective cohort data on this topic. But we only have we only have a self-report data. So there might be misclassification of the exposure. And another limitation in our analysis is related to the heterogeneity of the infection variable, because different cohorts using different tool or different question to collect the data. Some of them are quite different, and sometimes it is difficult to harmonize the infection variable across the 6 cohorts. Another limitation is, although we have combined data from 6 cohorts, but the number of cases are still not large, so we could not do a lot of stratify analysis to investigate the effect modification. And as I mentioned, we only have self-report data, and we can only assess the infection exposure in general. So next step, I think we need to investigate the effect of specific pathogen. And another extension could be using medical data to assess the infection exposure. Another possibility is to use the treatment data or vaccination data to test our hypothesis. If maternal infection is related to childhood leukemia, treatment or vaccination might modify the risk. Another important direction is related to the basic science, because actually we don't know the exact mechanism under this association, although we have some hypotheses. but the evidence, we don't have clear evidence on this point. And I would like to thank my supervisors and colleagues in Oxford. And I also would like to thank my collaborators in I4C in Denmark, Norway. And I also would like to thank my funding body, which support my PhD study in Oxford. And that's it. I'm happy to take your question. Thank you.
I say thank you again to you, Milena, for the invitation. And I found the previous presentation very interesting and inspiring. I wish I'd seen it before I presented the slides. So there is a slight, probably disjoint at the beginning, but I think the story will fit in a second. So I will be talking about mediation analysis in life course epidemiologies. And I don't really need to sort of tell this audience that it's broadly recognized, or that many chronic and recurring conditions, chronic such as cancer, are likely to be shaped by developmental processes experienced in utero, as we just discussed, at birth, for example, cesarean section was mentioned just now, and in early and later life through, for example, exposure to childhood infection. And these processes are complex and involve interconnected exposures. And this small diagram I'm showing here is like a historical representation of the debate about how to represent life course epidemiology problems. It's taken by a publication by Ben Schlolo and Ku in 2002 and represents the relationship between exposures in early life to respiratory infection and their association to lung function in adult life. It's obviously not relevant directly to this talk, but what is relevant in this graph is that it represents the many pathways that may link an earlier exposure to a later outcome. And this path can be biological, environmental, social. And some of these exposure and these pathways obviously consist of variables which are evolving over time. And the challenges, if you want to analyze anything from a life course perspective, are really considerable. Now, moving on to sort of the topic of this conference and the example we have just discussed from Jeff, if we are thinking of life course exposures leading to risk of childhood cancer, we may think of different periods of life in fetal life and in early life. And here is a very simple representation of this, with fetal growth, an earlier exposure followed by possibly a later exposure in childhood to infections, representing the delayed infection hypothesis that Milena mentioned. And of course, earlier, before the fetal growth, there could be maternal factors that will influence earlier on these two exposures I'm representing here. So this is a very simplified representation, but what sort of helps me discuss is that if you want to study complexities such as this, you may want to compare whether it is the earlier exposure or the later exposure that matter most. And this would, in the literature, be labeled as critical period models, or you may think that both matters, and that would be sort of a cumulative exposure model, or that one is stronger than the other, and those would be sensitive period models, or that they interact with leading to a pathway model. But the problem is we may hypothesize in which of these models is most logical, but if we want to sort of empirically validate our hypothesis, we need to have methods that allow us to distinguish these different explanations. And if we really want to do this, I think it's quite obvious from the drawing in front of us that we are asking causal questions. And in fact, we're asking questions to do with mediation. We want to see whether how a hypothesis that fetal growth causes an increased risk in childhood cancer, we may want to try to explain that as a causal association by understanding its pathways. And one possible pathway would be early life or in utero exposure to infections. So mediation analysis, I think, is the way forward if we want to understand mechanisms once we have sort of hypothesized a possible cause of an outcome such as childhood cancer. And if we work in mediation analysis, we probably have, if any of you works in this, we may have been faced with a choice of using two possible approaches to the analysis of such data. One is the most traditional strand of path analysis, and the other one is what I call counterfactual based causal inference. It seems sort of a mouthful, but really tries to discriminate between the two because there is obviously an overlap between the two. But the path analysis is the most traditional methods used mostly in psychology, and has many limitations, mostly that it relies on relationships between the variables in our diagram, which are linear, and mostly allows you to deal only with continuous outcomes. But as in this example here, we have childhood cancer, which is probably a time to event outcome. Early life infection could be a binary variable or a time varying binary infection. And with path analysis, we can't really deal with such complications. In addition, with path analysis, we don't have a very formal definition of the mediatory effect. In contrast, counterfactual-based causal inference uses a more formal approach to define the mediatory effects. Not only that, but also has the generality to deal with all possible types of variables. And it does that by sort of relaxing the link to modeling and thinking instead of comparing alternative hypothetical worlds. So my talk now will consist of two parts, really. I'll give a brief introduction to definitions of mediation effect using the counterfactual-based approach, and then work through an example. Unfortunately, my example is not to do with causal inference, but I think will be sufficiently linked to give you an idea of what you could do with an outcome which is cancer. Sorry, I should have said a cancer outcome. So the counterfactual approach, what we want to do, as I said, we want to compare our alternative hypothetical worlds. And now my representation, instead of having specific variables, talks about a variable A1, which is earlier in the life course, and a variable A2, which is later in the life course. And we want to separate and understand pathways linking A1 to Y. And we do this by invoking certain potential outcomes or counterfactuals. We first will introduce the concept of Y A1, little a1, and this is the potential outcome had we been able to intervene on the earlier exposure A1 and set it to a particular value, like no maternal infection. And then Y A1 represents that potential outcome, that potential cancer, yes or no, later for the child of that mother. We can do the same for the intermediate exposure A2, and we call that the potential outcome had we been able to intervene on A2. And we could also consider a potential outcome where we intervene on both exposures. So we hypothetically set maternal infection to be 0 and childhood infection to be 0, for example, and see what that potential outcome is, and then a combination of the other values for A1 and A2. There is one last potential outcome I want to introduce you to, which is, in fact, a potential mediator, which is what is the value of the intermediate exposure that we would have seen had we been able to intervene on A1. So having defined this concept, I'm now going to define the effects, the mediation effects that we will consider in a moment, but starting first with the total causal effect of A1, our earlier exposure. So the total causal effect of A1 can be seen as a contrast of averages of these potential outcomes. So this is the average of the potential outcome had everybody been exposed to A1, say all mothers had been exposed to infection, versus the average potential outcome had none of the mother been exposed to exposure. Having understood this, we now start to separate the pathways which involve and do not involve A2. And the one sort of bunch of sort of mediation effects which are often used are the natural effects, and this is what I'm defining here. So the natural direct effect is again represented as a contrast of means of potential outcomes, but these are sort of quite complex potential outcomes. So the one on the left is a potential outcome when we were hypothetically had intervened on all mother and exposed them all to infection, while we were holding the second level, the intermediate exposure, to take the value had there be no infections for the mother. So this is the potential mediator without exposure. And we contrast this potential outcome with one where the intermediate exposure is left to take the same value, but the earlier exposure is set to be not exposed. So we are really picking up the effect of A1 on Y, which does not involve A2, because A2 is held constant on both sides of this contrast. You can see that you can do the opposite and define the natural indirect effect when you hold the value of the first exposure while you switch on and off the second exposure in terms of whether or not there was an exposure to A1 or there was not an exposure to A1. So this captured the effect of A1 on Y transmitted via the intermediate exposure. These are beautiful definitions and many people have used them, but they have a major limitation in life course epidemiology, because for estimating, and I'm not going into the details of how you estimate these effects, you need a very strong assumption. You need the assumption that there is no, what we call no intermediate confounding, which means that there is no other pathway involving A1 that also influences how A2 affects Y. So in terms of graphical design, it would mean that there is no other variables downstream from A1 which influences A2 and also influences Y. And in life course epidemiology, you have that earlier exposure will influence, for example, dietary, the diet of the child, or the exposure to school of the child, that will influence whether they get infected or not, and whether they have the outcome of interest. So this sort of puts us in a very difficult situation. Beautiful definitions, but we can't really estimate them in life course epidemiology. Luckily, there is an alternative way of defining mediation effect, and I'll stop here now. This is my last technical slide. And they have got a funny name of randomized intervention analogues of the natural directed indirect effect. And what they do is, instead of thinking of hypothetically intervening on A1, hypothetically intervening on A2, we simply say, what would have happened if we could change the distribution of A2? And this is what this graph represents. So we could, again, we do a mental exercise and we think, what would have happened if we could have a control on the distribution of the intermediate exposure, not the actual value of each individual, but the distribution in the population, for example, with a public health policy. And if we could intervene on that, could we find an equivalent of direct and indirect effect? And luckily, these definitions do not require this assumption of no intermediate confounding, and therefore are very useful in our context. Another nice feature of these analogues, in randomized intervention analogues, is that they have been generalized to setting with multiple mediators. And this is one where I will move on to my example to show that in most situations, and in the situation that Jeff was considering, maybe you don't just think of one possible pathway from maternal exposure to infection, but you may think of many possible pathways. So this brings me to an example which arose from my collaboration at the Institute of Child Health at University College London, where I work. It's a collaboration with psychiatrists interested in eating disorders in adolescents. So there is plenty of evidence that body size throughout infancy and childhood may be influencing the onset of eating disorders in adulthood, in adolescence. And not surprisingly, the beautiful ALSPAC study that contributes to your consortium also has wonderful data on psychological outcomes in adolescents. So and as well as anthropometric data, which are relevant here. So the outcome that I will discuss is a score which is standardized, created from questionnaire responses by the participants when they were about 13 years of age. And from this score, we defined a binge eating indicator, which was obviously in response, which is an indicator of eating disorders risk. And the exposures that we will consider here are so the trajectory of growth from birth weight to various size in BMI. And the measurements we are considering is from age 7 yearly, roughly yearly, up to age 12, so up to before the measure of the outcome. So here is a representation of, so first look at the data when we just look at 2 time points of the exposure, just to reflect the examples I've just discussed. So we have birth weight as the equivalent of A1 and BMI at age 12 as my A2. And here just to highlight that this is a typical case where you would have intermediate confounding, because obviously earlier body mass index size will be downstream from A1 and influence A2 as well as the outcome. So let's first look at the association between birth weight and this score. The score, I said, is standardized, so it's the results are in terms of standardized units of this score. And the total association between birth weight and binge eating is quite small. It's 0.5 of a standard deviation, but it's significant. But then let's try to examine how much of that is mediated through BMI at 12. And I use interventional effects by imagining that I can shift that distribution of BMI at 12, for example, through a public health intervention. So I find that half of the total effect is not involving BMI at 12. You can say this is the direct effect, and it's 0.023. And how do I interpret this? So this is sort of the twist that we need to make when adopting these definitions. Shifting birth weight by 1 standard deviation while holding the distribution of BMI at 12 leads to an increase. So we are considering keeping the distribution of BMI as if there was no high birth weight. And the indirect effect is about the same size. And this is instead the effect of holding birth weight at the same level, but seeing what would be the effect by shifting the distribution of BMI at 12. So far, so good. But I have multiple measures of BMI, and why not considering all the information I have and look at them at the problem as a multiple mediator problem. And here I use again the interventional effects definition, and I imagine what would happen if I could shift the distribution of the child's size in between age 7 and 12, while to reflect what would have happened had birth weight been increased by 1 standard deviation or not, for example. And I find that the indirect effect, so the one represented by all the red arrows here, is actually larger than what I had before. So there is a greater mediatory pathways from birth weight to binge eating. But I don't want to stop here also, because now that I have multiple mediators, I may want to ask whether there is an early life or a later life greater impact. So now I can generalize and split the contribution and ask myself, what would be the impact of just shifting the distributions of the earlier BMI as opposed to the later BMI? And I find that actually concentrating on shifting these values in early childhood only practically has no impact on binge BMI. Note that, however, there may be an effect of the earlier BMI through sort of tracking through later BMI. So this arrow is not part of the blue pathway that I'm considering. There could still be some contribution through earlier BMI to later BMI. So early growth on its own from this conclusion does not seem to contribute. But now I'm going to criticize my work even more and say, well, all those measurements of BMI could be very imprecise and affected by measurement error. Why don't I instead extract the information I have on the trajectories of these children and ask the question of what are the features of those growth trajectories that matter? And here, without going into greater details, I have created some sort of latent dimensions from fitting a multi-level model on the repeated measures of BMI and extracted the concept of the size of the child, so the underlying propensity of being of a certain size throughout childhood, and separated it from the velocity of growth through which BMI advanced in childhood. And now I can, using, again, interventional effects, ask the question, is the pathways from birth weight to binge eating mostly via size or mostly via velocity? It turns out that it's mostly via size, not the velocity of growth in BMI. And this seems quite contradictory to my earlier results, you may probably want to ask me, because earlier I found that the BMI 7 and BMI 9 were not so important. But in fact, because there is so much tracking in BMI in childhood and in adult life, actually, it turns out that most of the causal effect of birth weight is really transmitted by your natural propensity of being of a certain size, or natural propensity or environmental propensity, I should say. So my conclusions from this exercise of mediation analysis, using a stepwise understanding of my data, is that really it's the latent size that is the greater driver. And this brings me to my conclusions, is that with this example and my introduction to counterfactual mediation analysis, I wanted to stress that there are ways of addressing the difficulties posed by life course epidemiology. Understanding the effects of lifetime exposure is the aim that we have, but our ability to perform such investigation is constrained by the often unrealistic assumptions needed to address them. Luckily, interventional effects and disparity measures, of which I haven't really spoke about, but I'm happy to answer questions about, have the potential of addressing questions which are not exactly the same as the natural effects that we would like to use, but they are very useful nevertheless. And including latent dimensions allowed us to address measurement error issues and, in fact, missing data issues that would otherwise might introduce biases in our conclusions. Thank you very much for listening. I hope I haven't gone over time. This is joint work with Andrew Pickles and Moritz Herl from the Institute of Psychiatry.